I call upon Mr. Balagopal Chandrasekhar, founder, Terumo Penpol Private Limited, as well as Professor James Spudich, Professor of Medicine, Stanford University, to come over to the stage and take their seats in the dais. This session is being chaired by Mr. Balagopal Chandrasekhar. Let me introduce him. Mr. Balagopal was captivated by a newspaper article on blood bag technology, and he embarked on a journey that led to pioneering steps in biomedical device manufacture. I welcome both Professor James Spudich, who is the chief speaker of this session, as well as Mr. Balagopal Chandrasekhar to this session.
Yes.
And I request Mr. Balagopal Chandrasekhar to chair this session, introducing Professor James Spudich.
Good afternoon. I think I'll go plunge as quickly as possible into the introduction, but I want to do justice to the speaker who you are going to hear now. Give me about 3 to 4 minutes for saying something which will give you some idea about whom James Spudich is and the kind of work he has done. It gives me great pleasure to say a few words about James Spudich, an outstanding scientist, world-renowned researcher and teacher. But let me start with a story, because I think that will tell you a lot about him. The room was packed with a standing room only crowd. The year was 1963, and the setting was Woods Hole, Massachusetts, renowned for its summer classes in biology. James Spudich was doing his Bachelor of Science in Chemistry, had just finished his bachelor's degree at the University of Illinois, and he stood at the front of the room wiping the blackboard in preparation for the next speaker, who was Professor Woody Hastings. But as Hastings stepped up to the lectern, the professor suddenly threw a curveball to the crowd. He introduced his young student, James Spudich, who strolled up to the podium and began giving the lecture in Hastings' place to the shocked audience. Nobody was expecting it, Spudich recalled. Only he and Hastings knew the secret, and the crowd responded enthusiastically. But for Spudich, even more important than impressing the audience was impressing a young lady at the back of the room whom he had his eyes on, and his lecture about his work did the trick. They soon began dating, and 1 year later they were married. Please acknowledge Anama Spudich, who was the one who was responsible for this young chemistry graduate giving this lecture in front of an unsuspecting audience. Professor Spudich has made his mark with pioneering discoveries on molecular motors, the tiny powerhouses responsible for movement at the cellular level. For this research, he has won a 2016 Alumni Achievement Award, one in a long line of honors. I will not read out all the honors and the decorations and the lectureships and all the other glittering milestones in an outstanding career. Professor Spudich grew up in a small coal mining town in Illinois, about an hour south of Springfield. And he and his brother they had a lot of adventures, which also I think if you Google, you will read about how they almost blew up the neighborhood during one of their chemistry experiments. Anyway, luckily, they didn't blow themselves up, because James Spudich went on to become a world-famous biochemist and scientist, and his brother went on to become a famous mathematician. Then, as I mentioned, I'll just go very quickly to the... So, to do his work on molecular motors and related areas, In 1994, Professor Spudich's team, they built a laser trap with the help of Steve Chu, a Stanford physicist who won the Nobel Prize in 1997 for trapping atoms. The collaboration between Professor Spudich and Chu was unique. Spudich had his team work part of the time for a year or more in Chu's physics lab, while Chu's physics students spent part of their time and several years in Professor Spudich's lab learning biochemistry. This student-swapping collaboration inspired Spudich and Chu to bring a new idea to Condoleezza Rice, the former Secretary of State and then Provost of Stanford. They suggested to her the creation of a program that would bring together talent from physics, biology, chemistry, engineering, computational sciences, and the clinical sciences. They called it BioX. Professor Spudich served as its first director from 1998 to 2001, and today over 600 faculty members across campus are part of the BioX program. Interestingly, apart from all his outstanding achievements in academia and in science, Professor Spudich also started his first company in 1998, developing a drug that makes the healthy part of a damaged heart work harder so that overall heart function remains the same. A second drug aims to improve muscle function for patients with ALS. Both are in the late stage of clinical trials. Meanwhile, a second company started in 2012 is developing a drug that slows down a racing heart in patients with a mutation that causes their heart muscle to overwork. One of his companies, of which Professor Spudich was one of the main promoters, was bought by Bristol Myers Squibb for, I understand, $13 billion sometime. I mean, that's what Google says, Anna. So I feel that for a seminar like this, which is being arranged, organized on translational research, to get across the idea that the fruits of research must reach society and must serve the people. I believe Professor Spudich is the most fitting person to speak at a seminar on translational research in biomedical sciences. He combines his vast and varied experience in biophysics, biochemistry, genetics, and structural biology with a deep interest in seeing that his scientific findings are applied in the development of better ways of understanding medical problems and in devising better solutions. Please join me in welcoming Professor James Spudich to deliver his talk.
I have never had an introduction like that. And stories are all true. I can't imagine where you found them. Unbelievable. So I've cut down my slides to the bare minimum because I knew the timing on the schedule was going to have me right up against lunch, and that's a problem. And so I cut out a lot of data slides. You can go back and read papers. I'll show a little bit of data, but not very much. But I wanted to get the message across that if you want translational, if you want to develop a society for translational research, then you have to invest in basic, very basic research. My research, I'm trained as a chemist initially, a chemist, physicist, and had very little biology courses, actually, until Woods Hole, where we met. And my work has been very fundamental, and as you'll see, I'll give you a little bit of that data, having to do with very, very basic understanding of what the contractile system does in cells. And I'm going to tell you the personal story of how that basic research led to the biotech companies and then to medicines. So that's the lesson today. So when I started my laboratory many, many years ago, people did not know how all of these kind of movements you're seeing in this slide occurred. Muscle had been studied, and myosin, the molecular motor in muscle, was known, although it was not known how it really worked. But it wasn't known whether there was myosin in non-muscle cells. So one of my starting projects, which has been consecutively funded for more than 5 decades by the NIH, has given total support to very, very fundamental research, was to understand how do all of these movements occur. In the lower right, of course, you see a beating cardiomyocyte. That's what causes your heart to contract. But in the upper left, you see a cell migrating over surface. In the middle, on the upper panel, you see a cell dividing into 2 daughter cells. All of these kinds of events require a motor protein that can cause this kind of force production to cause these kind of movements in cell division. And the lower left is plant cells with cytoplasmic streaming. So life is movement, and it's driven by molecular motors. And since I started my lab, my own contributions, plus a lot of my friends and colleagues in this field, have shown that, in fact, myosins, but also another molecular motor called kinesins, are driving these movements. But how does it really work? And that's what the basic research has been on my part for many, many years. So I'm going to tell you about one myosin type. They're different myosins. They look a little different, but they pretty much look like this. They have 2 heads with a coiled tail. And these movements and force production are really driven to a very large extent by this particular molecular motor, which is my favorite enzyme in biology. This myosin has to bind to another protein called actin, which makes these filaments colored in blue here, in order to produce the contraction. Okay, so it's an actin-myosin interaction driven by the hydrolysis of ATP, which is the fuel that drives the contractile event. Now, in early years, we studied an amoeba, as I mentioned in my little preface. It's called Dictyostelium discoideum. It's an amoeboid cell that actually looks very much and behaves very much like a leukocyte in your body. So we were studying it as a model system because we could manipulate it by genetics and structural biology and biochemistry. And in this particular experiment, we genetically removed the myosin from this cell to see what the cell would not be able to do. And of course, it wasn't a complete surprise that when we eliminated the myosin, the cell could no longer divide into 2 daughter cells. Because as I told you, we knew you needed a molecular motor to do that. By the way, we discovered myosin, that myosin exists in this cell. Nobody knew that before. Having showed that we provided the genetic proof that you needed this molecular motor for cell division, we were then able to use molecular genetic methods to put in myosin back in using a plasmid. You can put it back into the cell and rescue the cytokinetic event. And then you could start making mutations on the myosin and see how you can modulate it and still rescue, or whether the mutation failed to rescue. So there was a lot of work of that kind being done. This particular experiment was simply to put a fluorescent tag on the myosin molecule so you'd be able to see it in the cell. And when you put the fluorescent tag on, you can then watch where the myosin is in the cell and where it's going, and you can see it accumulates in the middle of the cell. It forms like a little muscle that's transient, and that divides the cell in two. So there's a decade of work with, I don't know, 50 to 100 papers describing much of this work. But this was very, very fundamental research. And at this time, if you told me that I'd someday be starting a biotech company, I would say, that's highly unlikely. But this is the deep understanding from this model system that we got was essential to really go to where I'm going to take you now. Because having this deep understanding, there were textbook models of how muscle contracts. But as you all know, in the textbooks, those are largely models. There's often not a lot of data that proves the model. It's very hard to prove a model. You can, mainly in the laboratory, you try to disprove your model. If you accumulate enough vast evidence, then you can pretty much believe the model. When I entered this field and looked at the muscle, there was a lot of confusion. Even though people thought they knew how it worked in the textbooks, there was a lot of confusion about what was really going on if you were in the muscle meetings that we all attended. And what was missing, I thought, being trained as a hardcore biochemist, was you need to be able to reduce movement and force production, which is so important for muscle contraction and all these non-muscle movements. You need to be able to reduce those to purified proteins. So you need functional assays for movement and force production. And it took us many years. It turned out to be very simple once you knew how to do it. But we reconstituted with purified actin and purified myosin a motility assay where we could watch the proteins move in relation to one another. And one trick, the actin filament is only 9 nanometers. Nanometer is 1/billionth of a meter, so it's tiny, tiny size. So you can't really see an actin filament without doing something. But you can put a fluorescent molecule on the actin, and then under a good fluorescence microscope, you see what you see in the black square here. You can see individual actin filaments which have landed onto a glass slide where we have placed a lot of these myosin molecules, which you can't see because they're not labeled with fluorescence. But the actin goes and it binds to those myosin molecules. And nothing's going to happen because there's no fuel. You need some chemical energy that's going to get transduced into mechanical motion. So when you add ATP, what you see is this. And this is almost a real-time movie. So you can imagine when my student, Steve Crone, first got this to work and ran into my office with such excitement. I can't tell you what he said. It was a 4-letter word. Very excited he was. And it's a robust assay where you can actually measure the velocity. Turns out the velocity is very close to the maximum velocity you can contract your muscle, or the maximum velocity that's occurring when your heart is contracting. And so it replicates that. So this was fundamental and really changed the whole field, because suddenly you could study the motion with a quantitative assay. But how about measuring the force? And so this was already mentioned to you. What we had to do was to combine physics with biochemistry, because in this case, we needed to use the physics of laser trapping. And as you were told in the introduction, Steve Chu, who was a physicist trapping atoms, for which he got a Nobel Prize in 1997, had also discovered accidentally that you could trap larger particles. And so I went to talk to Steve, and he agreed to help us to build a laser trap, which was built actually by my student going over to Steve's physics department. And as you were told, this led to the development of BioX, which was very exciting for Stanford and a lot of other places that have replicated that approach. But what we were able to do is to use laser traps, shown here in red, focusing them onto 1 micron polystyrene little plastic beads. And when you focus a laser trap on a particle like that, it actually traps it in space. So if you move the laser beam around, you can move the particle around. And by putting a little bead on each end of a single actin filament and having 2 laser beams, you could take the actin filament and move it around and put it on top of this bump, which is on a glass slide, and that's just another plastic particle, where we've managed to put a single myosin molecule. We were driven to have to do this incredible experiment because without doing this experiment, the field was at a standstill as to how this motor really transduced the chemical energy into mechanical motion. There were widely different views at this point. And this was the experiment that really showed what's going on, because it demonstrated that you get a very small step in motion, and we were able, using this single molecule approach, to actually measure the force a single molecule can produce. And that force is 2 piconewtons, really, really, really tiny, but you have billions of myosins in your heart. And so when you take 2 piconewtons per myosin molecule, it actually adds up quite accurately to how much force you need for your heart to function. So lots of academic studies for many years were really essential to set the stage for translational research. And about a dozen years ago, we decided to focus all of our efforts on studying a disease called hypertrophic cardiomyopathy, which is a big problem all over the world, certainly here in India as well. And it affects like 1 out of 200 people. That means someone in this room undoubtedly has it. And it's the most common cause of sudden cardiac death in individuals under the age of 35. I'm sure some of you in the room know someone who died that young suddenly, maybe without even realizing what they had. Probably they had hypertrophic cardiomyopathy. It's a genetic disease, and about 1/3 of the genetic mutations that cause hypertrophic cardiomyopathy result in mutations in this myosin molecular motor, which is operating in your heart. It's called beta cardiac myosin. Okay? Importantly, those mutations are causing the myosin to be too good a motor. You know, it's doing too good a job. The power output that this myosin is producing is more than you want it to be. So your heart is hypercontractile. I like to say it's as if you were out for a run when you're just sitting here listening to my lecture. So your heart is overworking, and your body thinks you're exercising, so your heart starts to hypertrophy, which is what happens when you go out and you go for a run. If you're an athlete, you have a larger heart than the rest of us. And your skeletal muscles also, of course, increase. That hypertrophy, if you stop exercising, will go back down. So that hypertrophy is normal if you're exercising. If you're carrying this mutation, it's as if you're exercising 24 hours a day, 7 days a week. You're never stopping. Your heart continues to hypertrophy, and then you get HCM, shown here schematically, where your muscle in your heart has hypertrophy to such an extent that your left ventricle is diminished in size, is no longer able to push enough blood through your body, and that's a very serious stage of the disease. So, again, if you want to develop a small molecule therapeutic, which is the story that I'll finish with, you need to understand what these mutations are doing to the human cardiac myosin that cause it to be a better motor. Better in the sense that it's producing more power output, not better for your health. Okay, so here's where I just took out all my slides in order to save time. Here's the answer. There were many possible explanations for what these mutations were doing, but the answer turned out to be totally different from what we and everybody in the field thought it was going to be. And it turns out the answer is that as you're sitting here, about half of your myosins in your heart are in a state shown in the upper right, where the two heads are not sticking out, where they can interact with actin and produce force, but the two heads are folded back onto their own tail, so they can't interact with actin. And half of the cardiac myosins in your heart are in that state. If you go out for a run, you have normal physiological responses that alter the myosin molecule, and more heads come out of that state and interact with actin. And so this is your normal physiological way of increasing your power output just physiologically. But in the case of HCM, it turns out, and I'm not showing you any of the data, you can read the papers, to all of our surprise, it turned out that what HCM mutations are actually doing are causing more of these heads, which are normally, like half the heads are normally in this off state, it's causing them to open up and interact with actin, and therefore you have a higher power output. So that's about an hour's seminar worth of data, which I'm not showing you. But understanding this, one can then try to think about, well, if that's the case, why can't you develop a small molecule that will bind to that myosin directly and cause it, if you can, cause it to go back into the folded state and just reverse the process? That's as good as you can do in the world of translational research. You have a myosin molecule where you know it's mutations in that molecule that are causative of the disease, and now you're going to try to make a small therapeutic agent which binds directly to that defective myosin and basically reverses the hypercontractility back to normal. That was the goal, and it was with that goal we started a company called Myocardia. Now, let me emphasize with this slide that before Myocardia, there was no treatment other than if you get to a severe stage, you need, the only thing you can do is open heart surgery and myectomy to cut away some of this excess muscle. There's another approach where you can inject alcohol, ethanol, into the heart, causing a heart attack with some of that dead cardiac muscle then being eliminated. But both of these are horrendous and not really treatments you want to do. And this was all that was available. 1 out of 200 people are suffering from this. So we then started Myocardia and developed a small molecule called mavacamten. This is its structure. Just a brief, for those of you who don't understand what's involved in drug discovery, it's very, very hard. You have to have an assay like the actin interaction with myosin, could be the assay I showed you, the motility assay. Or it can be the fact that actin activates the myosin ATPase, and you can use that in a screen of hundreds of thousands of small molecules. And you look for what are called hits. That is, you look for a few molecules out of that library of molecules that actually, in this case, inhibit the actin-activated myosin ATPase, or the velocity, or whatever your assay is, to reduce the power output back to normal. And you find such molecules. Typically, you find one out of about 1,000 small molecules are hits. And then you're at a starting point, because you have to make that small molecule work in a potent way, much more potently than the hits typically are. And furthermore, you don't want that molecule inhibiting skeletal muscle or smooth muscle. You want it to inhibit only cardiac myosin. Cardiac and smooth and skeletal muscles are 95% identical. But that 5% difference enabled us to make a cardiac-specific drug. So you need potency, you need specificity, you need permeability, you need solubility, you need stability of the drug. All of these things are a very hard process, which involve a lot of chemistry, dozens of chemists working full-time, doing what's called SAR, structure-activity relationships, to increase the potency and so forth, as I said. All right, so that's what you do. And it turns out that what mavacamten does, which you can only discover by going back to basic research. So my laboratory at Stanford collaborated with MyoKardia, which is just 30 minutes away in the Bay Area, to carry out a series of studies. And those studies showed that what the mavacamten does is basically reverse what the HCM mutations do. So the HCM is putting more heads in play so that you are hypercontractile. And if you use the right dose of mavacamten, you can take those excess heads that have been put into play, probably not the same ones, but that number of heads out of play to reduce the power output back to normal. And the hypothesis was that would very quickly help patients. The further sequence involved in the drug discovery is you got to show it works in cells, you got to show it works in animals, and then you have to go through phase 1 toxicity studies, phase 2 efficacy studies, and finally phase 3 studies. And most drugs fall out along this way. So it's a really tough process. MyoKardia took this all the way through phase 3 clinical trials. The company was founded in 2012. 8 short years later, phase 3 clinical trials were completed that looked spectacular. So spectacular that Bristol-Myers Squibb just bought the company. And Bristol-Myers Squibb immediately got FDA approval, and on the market for the last 6 months or so has been mavacamten, which somebody gave the name Camzyos. I'm not sure how that happened, but anyway, I guess it's catchy. And this drug, which is a drug now, is available. I know that I'm aware that Bristol-Myers Squibb is negotiating with India about getting this drug into this country, but it's being used now in the United States. I have one personal testimonial that is from one of the very first patients treated with this. Her name, Charnel Mumford, 67-year-old woman. She was getting treated at Stanford. And within a few weeks of starting this therapy, she said, I noticed little things at first. Like one day I was able to walk up a hill which she had not been able to do, increasingly hard to do for her. But she noticed she was doing that much more easily. And then not long after that, she's now walking marathon, okay? And so this has been transformative for her. And I hear more and more stories of these kind, which is incredibly satisfying. Furthermore, I'll tell you that patients who are being treated who are already hypertrophic, as she was, we already are seeing within a few months that the hypertrophy is decreasing, which I'm not surprised by, because as I told you, the hypertrophy is a result of being hypercontractile. Your body thinks you're out for a run, so your heart hypertrophies. If you stop running, the hypertrophy decreases, and that's exactly what we're beginning to see. So, conclusions. Myosin is the most beautiful, important enzyme in biology. I have no prejudices about that. It's involved in all of these different biological functions. Really fundamental, basic understanding of the molecule was really established in academic studies, in my laboratory and others, without any idea in the early days that this was going to lead to drug discovery. So you have to invest in really hardcore basic research if you want translational research. This understanding made it possible for clinical studies in biotech environment, which gave rise to this drug. And with that, I'll just thank current and recent members of my lab and others from Myocardia who I don't have on this slide, collaborators, unending funding from NIH for 5 decades. I still have grants from them working on hypertrophic cardiomyopathy where there's still more to do. With that, I'll say thank you.
I think we have time for a couple of questions. I'm sure you must have all been as fascinated as I was by, of course, I would have understood much less than all of you about what Jim Spudich was talking, but I'm sure some of you will be having any questions. We can probably take 1 or 2 questions, and the rest you can probably ask him in person during the lunch break. That's right. So if there's...
I think if I heard you right, you're asking how many molecules did we have to go through with the SAR to get mavacamten in the end? Okay, I mean, typically in drug discovery, it can be thousands. You might, I mean, my wife and I have founded another company, Kinomics, actually targeting myosins in parasites for malaria treatment. And I can tell you we've already, in our SAR in that company, we've already made over 1,500 derivatives, and we still don't have a molecule that we think is close to being a drug. So it takes a very long process. And it was quicker in the case of mavacamten. The initial hit already had pretty good selectivity for cardiac. We were lucky. It wasn't totally selective. That had to be improved. It was already like the potency was already better than typical. And the number of changes on the molecule that we had to make mavacamten was really minimal. So in that case, it was only something in the order of less than 100 different changes that had to be tried before we got the molecule we wanted. Oh,
Or is it a particular, because we do this molecular docking and simulation studies,
ah,
are we supposed to retain the interacting atoms and then do the modifications, or are we, is it okay if we change the interacting atoms?
sure. Well, so you bring up a very important point, which of course I completely eliminated, but in all of the companies that I've been involved in, the structure is really important. So we know where this mavacamten binds, we know the pocket, and we knew that even with the earlier hit, okay? So using modern AI approaches is really important in structural approaches. But what you do is you look at not mavacamten, but an earlier precursor that's bound to that pocket. You look at, see what the interactions are, and you look at neighboring residues, of course, and then you can predict. Now, the pocket's only so big, so there's no point in putting some big structure onto the molecule. It's not going to go in. So both size and then where you have hydrophilic versus hydrophobic side chains become sort of obvious from the structure. So you go back and you modify according to that knowledge base. I think that's what you were getting at. Maybe one, oh, my wife has something she has to say.
Was already so well defined, the three-dimensional structure
Of the myosin.
of the myosin molecule, so that you had a view of how the molecule is basically structured and where the pockets are.
It's very hard to do drug discovery in the way I described without having a high-resolution structure of the target protein of interest, because you really need to be able to do this SAR using that structural information. It's not always necessary. There are a lot of drugs that did not go through that process, but it's a lot harder, and your success rate is much lower.
Drug discovery as well as, phenotypic screening in some kind of drug discoveries. So
Turn on the mic and say what.
no, I was saying that your approach was targeted drug discovery that was directed at a molecule
Yes.
that you understood, whereas in other cases there is of course phenotypic screening,
Yeah.
which does not require the information about the specific molecule that you're targeting.
Right. But the success rate with phenotypic screening is much, much lower.
I think we can probably have two more questions. This is the second last and
Or
last question.
I'm happy with just one more, and then we can talk at lunch.
Yeah.
Everybody's tired, I'm sure.
Thank you for your wonderful presentation.
Where are you? Ah,
You very well explained the importance and relevance of basic research. And as a young faculty in Kerala University and India, we are like, now we are little bit tensed what kind of research we are doing. Everybody is pressurizing us to do the translational research. And we don't have getting enough time to do the basic research. And what is your message regarding that? And how we can probably,
yeah.
the funding and everything is now related to translational aspect. And
Yeah.
another thing, what are the roadblocks you are actually faced to approach a company? And suppose we have a product in our, or a smart molecule in us, and how we can actually approach a company effectively. And then you are actually develop a screening system in your own lab. And how effective a screening system developed in your lab accepted by others in the scientific scenario?
Okay, that's 3 questions.
So this will be the last question.
But
You'll have to ask during the lunch.
But let me just say, your first question about basic research, that's why I went out of my way to emphasize the understanding your target is, if you look at statistics in drug discovery, gives you a much higher probability of success. So And as I described in my case, understanding the target really involved fundamental research in starting with studies with amoebae. And so I think there's no question that it's very difficult to just plunge into heart and say, okay, I want to reduce power output. So how are you going to do that? You need to understand that molecule in real detail. There was no way to understand human cardiac myosin in that kind of detail. You couldn't get it to study. So that understanding came from other model systems. So very basic research is very important to support by whatever government is supporting it, no question about that. None of this screening was done in my lab, okay? The understanding that I described of how the molecule works, the development of the laser trap, motility assay, so on, done in the lab. But the screening is done by the company. And in the case of myocardia, it's not even done in-house because you can do that in India. The company that my wife and I now have, Kinomics, we have 12 full-time chemists. They're here in India. So we tell them what molecules we want them to make. They make them and they send them to us. And then we study them in our Kinomics lab. Your third question was about if you have a small molecule, how do you get it the interest of a company like, say, Merck or Glaxo, or you want to, if it's for heart failure, maybe it's Novartis. So if you develop something like that in a small biotech company, the big company is going to be mostly interested in it if you've de-risked it, meaning you've shown that it's been through all of these permeability issues and stability issues, and it's ready to go in the clinic. And it actually works in animals. If you know that from the biotech company, then a big company would be interested in making a deal with you. In this case, Myocardia, the CEO said, no, we're not going to make a deal at the lead stage, so you can take it into phase 1 clinical trials. We're going to take it into phase 1 and 2 and 3 ourselves as a small biotech company. And then, of course, it's worth a lot more to the big company if they want it. And that's why they paid $13.1 billion to buy Myocardia. I don't know if that answers your question. Okay. Thank you for your attention.
Thank you, Jim. I think there was there are many points which he made, but one of the very important points for this conference, I think, which is very, very insightful, is the fact that there is a tremendous amount of basic research which precedes any translational work. And so put in layman's language, I think it would be that, it would be difficult to take a very targeted, I'm talking about from the science administration point of view, from governments and people who are going to fund this kind of research project. So it was very significant that Professor Spudich mentioned that the National Institutes of Health funded for nearly 5 decades. They have been funding his lab for the basic research which they have been doing. That is the key, that is the core. We cannot expect quick wins, and if we do quick wins kind of approach, I think we're going to be doomed to disappointment. We're going to be wasting a lot of money here. So I think that's been one of the most important points which he made today, which we have to have a kind of understanding that there is a lot of basic research involved. Because, for example, here, it was the basic understanding which came from all the research which he did for those decades, which made it very easy for him to decide on how is he going to go after a solution. What is he going to look for, isn't it? So I think you will all agree with me that we've been privileged to listen to an even though albeit very brief and very rushed, but in view of the constraints, unavoidable. I'll just end with this. There are many more things about James Spudich which you don't know. What you've seen is just like the tip of the iceberg. He's not an iceberg, he's a very warm person. So what I want to, I'll just quote from one thing which I picked up from the internet. As for James Spudich, he shows no signs of slowing down. But whenever he wants a break from this hectic pace, he goes to the air. He has been flying small planes for more than 40 years, and he even wrote a book on flying. Now he says, within inverted commas, you're 10,000 feet above the ground, and suddenly your whole perspective changes. You realize there are all kinds of things going on down there, and it puts perspective on the things we spend all of our time worrying about. As the great explorer, I can't pronounce his name, Antoine de Saint-Exupéry, expressed it, I fly because it releases my mind from the tyranny of petty things. Thank you, Professor James Spudich, for a wonderful lecture, and thank you all for having been a great audience. Thank you.
Thank you, Professor James Spudich, for your interesting lecture, and also thank Sri Balagopal Chandrasekhar for chairing the session.